cells, which of course we call a morula. And then by about 4 to 4.5 days, the ball is in the uterus, and at about 5 days, that little ball is going to implant, and we'll talk about that. So you start out 2 cells, 4 cells, and then a ball of cells. You just keep getting... Actually, it doesn't get bigger. It's like cutting up a pie. The pieces don't make the pie bigger, they just subdivide the pie. So at first, the cells really don't get bigger. But at 4.5 days, this cell, this ball of cells becomes hollow, and we call that hollow ball a blastocyst. Now that's not to demean it as being less than human, it just has a scientific blastocyst. And it is like a cyst. And it's this blastocyst that's going to penetrate into the wall of the uterus that you see in red up there. It's like planting a seed in the field. At about 5.5 to 6 days, the implantation, this is after ovulation, that implantation occurs. This is a miracle you're looking at right here. There's no way on earth that that blastocyst should implant in the uterus. First of all, it's a different person. It's a different tissue. It doesn't have the same antigens as the mother, no more than your son or daughter has exactly the same genes as you do and the same antigens. If you take tissue from your son or daughter and inject it in your body, you will build an immune response against your own children, against their tissue. And yet the uterus says, come on in. And it comes in and it's not rejected. It should be, but it's not. And if it were, that'd be the end of life. You'd never get another chance at that. That's irreducible complexity. This group of cells right here, that's the baby. You think, well, if that's the baby, what are these other cells that form the ring out here? Oh, are they interesting? The ones that form the ring are called the cytotrophoblast. I won't even bother you with this name, except it really gets interesting. The cytotrophoblast cells fuse together. So you got 2 cells, they fuse together, you get 1 cell with 2 nuclei. Then another one fuses, you get 1 cell with 3 nuclei, right? We call this a syncytium, so the name changes eventually to syncytial trophoblast. And it is these syncytial trophoblast cells that are implanting at about 6 days after ovulation that secrete a chemical called HCG, human chorionic gonadotrophin, and that's the chemical we use for determining whether somebody's pregnant or not. The little tests are looking for this HCG. So the earliest stage you could tell that somebody's pregnant would be about 6 days, right when the implantation is occurring. And these syncytial trophoblasts are the only cells that have the ability to penetrate and go into the uterus. Without these cells, it'd be the end. These are the cells that can penetrate through the uterine wall. In fact, these are the cells that are going to make the placenta. Now remember what these are? Syncytial trophoblast fused cells. The whole placenta, the whole surface of the placenta is 1 cell. About half the size of a basketball court. Biggest cell in the body. But it's really only about this big around. But it's folded and folded and folded and folded so that it gets all that surface into a little area. And it's all 1 cell, and it's seamless, and it separates mother's blood from baby's blood. But we're getting ahead of the story. Let's look at, there's the syncytium penetrating through the uterine wall. You see the little fingers going through. Here it's penetrated a little further, the blue and yellow up there, that's the baby part. Basically the baby is told, would you mind waiting a little while? You see, because it isn't going to do any good to work on you at this time. We need to create a plug-in. It's like having a nice TV, but if you can't plug it in, it doesn't do anything. And by the way, when you plug a TV in, is it simple? Is it just 3 holes in the wall? Is that all it is? You take and plug into any 3 holes and the TV runs? No, there's a power plant hooked on the other end of that plug. And the power plant's more complicated than the TV set. And that's the way the placenta is. It's one of the most incredible organs in the body. There's nothing else quite like it. I call it super organ. When I taught my medical students, they all knew I was talking about the placenta when I said super organ. And you need to make this whole organ, or you can forget about the developing baby. It's a TV with no plug-in. Now, birds and reptiles, they're like battery operated. They have an egg with yolk in it, and so they just use up the energy that's in the yolk, whereas we need to be plugged in. Now, the red you see down there, that's mother's blood vessel. There's a lot of blood vessels in the uterus, and they're going to be brought into close association with the green, which is the developing placenta, the fused syncytial trophoblasts. We're getting close. There's a mother's blood vessel there. And when that syncytial trophoblast hits those arteries, one of the most incredible things you can think about happens. The ends of the arteries of the mother's uterus get eroded so that the arteries just end, like cutting off a hose. There's about 20 arteries big enough that you can see them, maybe half the diameter of a lead pencil or so. 20 truncated arteries. Wow, you better have a nice seal on this placenta or this blood, which comes through at the rate eventually of a half a quart a minute, is going to lead to some pretty serious complications. But we need to get mother's blood up there to bring nutrients and oxygen to the baby when we finally get around to working on baby. This syncytial trophoblast makes a lot of hollow cavities inside. You can see it in the green here. And the blood is going to eventually flow into those cavities. See the red blood flowing into the cavities in the green syncytial trophoblast. Finally, you're going to have little processes all around the baby. And the baby's going to make blood vessels of its own, and they're going to be on one side of that syncytial trophoblast, that seamless cell layer, and mother's blood's going to be on the other side, and they'll never mix because the barrier between them is seamless. Eventually the placenta just becomes a flat disc, but early on here, for example, at 40 days of development, it goes all the way around, and if you'll notice, placenta is almost like hair. You see where the surface area is so big, you have all these little processes, each one of which has blood vessel inside from the baby, and then the outside of all this hairy processes is bathed by mother's blood. So there's a tremendous surface area for interchange. If you magnify just one of those little hair-like processes that are on the placenta, under the scanning electron microscope, you can see that these little processes have even tinier processes coming off from them, further increasing the surface area. So where you see black in this picture, that would be all solid mother's blood. The baby's blood vessels would be inside of those little finger-like processes, so the materials and nutrients have to go across to get to the baby. Let's cut a cross-section. See the red line? We're going to cut one of these little microscopic, tiny, tiny little processes and look at inside. If we look inside, there it is, the baby's blood vessels, but not the blood vessels of the baby's body so much as the blood vessels that are in the placenta. This is a little blood vessel, this is another one, this is another one. The red blood cells you see in here belong to the baby and were made by the baby. The red blood cells you see just out here are red blood cells of the mother, and between the two is this incredibly thin cell, which is that seamless syncytial trophoblast. The nuclei have the good discretion to get out of the way and move to the side to make very thin areas across which gas and nutrient exchange can occur. So everything going from mother's blood to baby's blood has to go across either passively or actively. For example, water could just go across passively. You don't need to pump it across. Sadly, alcohol will go across from mother's blood to baby's blood passively. So a mother who consumes alcohol will be giving the same dosage essentially to her baby. And a baby can be born with fetal alcohol syndrome. A mother who takes some of these recreational so-called drugs, sadly, many of these drugs go across the placental barrier passively. So you can have a baby born as a crack baby from crack cocaine. Oddly enough, most of the things that are really important to the baby have to be carried across with special molecules. For example, for the baby to make its own blood, it needs iron. The only way it can get iron is from the mother's blood, and the iron has to be transported across from the mother's side to the baby's side with special proteins, one of which is called transferrin, that can come over here, pick up the iron, carry it across, and release it on the baby's side. What a miracle we have here, just in the placenta. This is kind of what the placenta looks like in the hole. You can see a couple of the arteries that are truncated in there. And they're just blasting the blood out, like going up to some bushes. All those little villi we were showing you sort of look like branches of a bush with blood vessels inside. And the blood just gets sprayed over them, and it's sealed so it can't leak out of the sides of the placenta. The placenta is then really part of the baby, not part of the mother. But it's really not in the baby's body. It's attached by a fairly long hose. And you have to make sure you don't kink the hose. You know, ask any mother that's, she'll tell you when the baby was in the womb, they do loop-to-loops, and they do wing-overs, they do all these things. They do all this. How are you going to do that without kinking the tube? This is so gorgeous. There's a special jelly, not found anywhere else in the body, Wharton's jelly. The blood vessels are in there, and when you bend it, it resists kinking the blood vessels. Evolutionists say, what an incredible stroke of luck. Now, the most amazing thing I'm going to tell you all day is that when the baby's born, this placenta comes out. First the baby comes out, and then the placenta comes out as afterbirth, along with other membranes. And when that placenta unplugs, approximately 20 good-size arteries, virtually truncated at their ends, are now open to the lumen of the uterus to spray blood at approximately a half quart per minute. Do you think this is bad? You haven't heard the whole story yet. There is a mechanism in the placenta that suppresses blood clotting. Now, to get the picture, a hemophiliac, right, one that can't clot his blood, with 20 severed arteries, releasing blood at a little over a half a quart a minute. Anybody see a problem here? How on earth does anyone survive this? People have said this is the biggest wound any human being gets and lives to tell a story. Biggest wound. How bleeding to death is prevented is so amazing. When the blood vessel breaks apart, part goes with the placenta, part stays with the uterus. There's a little muscular sphincter, like a string on a purse, that, fortunately is on the side of the break towards the uterus, that squeezes suddenly and pinches off the blood, just sort of like a surgeon would use a hemostat to pinch a blood vessel. It all closes down so that under normal delivery you may lose a small cup of blood or something like that. Is this not amazing? Absolutely incredible. Flat out miracle. And yet people look at it and say, I don't see any design here. No design. What do you think? We give them a better example? We're not going to give them a better example. If they say, I don't see any design here, forget about it. You're not going to change that mind with scientific arguments. The only thing that can change that mind is the power of the Word of God and the Holy Spirit. That's the only thing you can do it. And lest you think it's not up to the task, it was nothing more than the Word that spoke the very universe into existence. Can that Word and the Holy Spirit change your hearts? Yeah, it can. It really can. Well, 4-week embryo, kind of interesting. Here he is in there, human embryo. Got the little yolk sac. The yolk sac, by the way, doesn't really make yolk. Blood vessels are developed in there in the earliest stages of blood formation. Let's get it out of the way. Evolutionists get really excited about these 4-week embryos. They say, look, it's got paddles. This is the fish stage. Oh, look, gills. I had a teacher, my daughter had a teacher in public school that said the baby uses the gills to breathe under the amniotic fluid. Wow. You can see the heart protruding there. The body's bent like a C because the nervous system grows faster than the rest of the body at first. And because it grows faster, it gets out ahead of the body. Notice the spinal cord has gone beyond the end of the body. That's because it's growing faster. If you look here, you see on the back, get my pointer up here, the spinal cord was growing so fast it got way out here and the body hasn't even kept up. There's those so-called gill slits. I can tell you 2 things about the gill slits that are absolutely true. You can bank on it. Number one, they are not gills. Number two, they're not slits. You get the idea? There are no gill slits. Now, what about that? Evolutionists look at that and say, there you go. There's your evidence for evolution. That's the tail stage. We have a tail. Well, as the baby grows, the body starts catching up, and what you see here protruding beyond the body ends up ending right about here in the smaller, just below the small of your back. That's where your spinal cord ends. And so then the body gets ahead of the spinal cord. It's not a tail. There's some little bones that develop down there at the end, and of course the spinal column has to end someplace, right? It is a linear structure, and there's a head on one end, and wherever the other end ends, there standing by will be an evolutionist pointing, saying, see, a tail. But how can you avoid having the other end? I tried this with a rope once. I had two ends, I cut off one end. Every time I cut off an end, I still had two ends. So Ernst Haeckel, back at the time of Darwin, talked about these gill slits being reminiscent of our evolutionary development, said the embryos in their development went through all the stages of their evolution, or at least many of them, and he showed how similar the chicken and the dog and the pig and the human were. But it turns out that these drawings he made were completely falsified. It was a hoax. The man was a charlatan, essentially. It's not like that at all. And yet, years ago, Dr. Spock, remember him? You couldn't have a baby without reading Dr. Spock. It was considered the handbook for babies, or the manual. And Dr. Spock, in his book Baby and Child Care, says each child as he develops is retracing the whole history of mankind, physically and spiritually, step by step. A baby starts off in the womb as a single tiny cell, just the way the first living thing appeared in the ocean. Weeks later, as he lies in the amniotic fluid of the womb, he has gills like a fish. Notice he even says we're retracing spiritually our evolutionary history. Well, this just simply isn't true. This has been known to be untrue for a long time. Here's a paper that was in a journal some time ago, and they pointed out that Haeckel's embryos are just simply a fraud. That if you look at